In today's video, we'll be studying Introduction to Histology. And here we have the outline of all we'll be studying: meaning of histology, tissue preparation, light microscopy, histological stains and dyes, histological sections, artifacts, and electron microscopy. Now, starting with the definition or meaning of histology, histology is simply the study of normal tissues. Okay, so the study of normal tissues. Now, the body is made up of cells that work together to perform a function as tissues, and these tissues work together to perform a function as organs. Organs work together to perform a function as systems, and so on. Okay, now histology is the study of normal body tissues. Now, we could also have instances where these body tissues are affected or distorted by certain diseases. Then we move on to the study of histopathology. Okay, so histopathology is the study of diseased tissues, is the study of tissues as they are affected by diseases and pathogens. Okay, now the next topic would be considering is tissue preparation. The next subtopic for this video is tissue preparation. Now, the particular technique of tissue preparation we'll be considering for this video is the paraffin technique of tissue preparation. And in this technique, we have about 5 to 6 steps, each of which we'll be considering one after the other. The first step in this technique of tissue preparation is tissue sampling. Now, of course, as I stated earlier, histology is the study of tissues, and these tissues are made up of cells that are majorly microscopic. Okay, the cells that make up these tissues are majorly microscopic. So it means in order to study the tissue efficiently, we need to study the cells. And in order to study the cells, we need to make use of a microscope, because these cells are essentially microscopic. Now, tissue preparation has to do with preparing these tissues or these tissue blocks so that they can be viewed under the microscope. And the first step we'll be considering is tissue sampling. Now, tissue sampling just has to do with obtaining the tissue itself. So, simply put, it is obtaining the tissue. Now, what tissue exactly are we obtaining? What we are obtaining is a tissue block. Okay, so we have a tissue block that could look something like this. Now, this tissue block is by standard about 1 centimeter in each of its dimension, and by tissue sampling, which is obtaining of the tissue block, this tissue block could be obtained by biopsy. Okay, it could be obtained by surgical excision or surgical cutting, excision, as well as post-mortem dissection. Post-mortem dissection. Now, looking at biopsy has to do with examining of tissues from a living being. Okay, so for biopsy, the tissue is obtained from a being or a human that is actually living. Okay, in order to examine, maybe for histopathological reasons and so on. For surgical excisions, the tissue block is gotten by means of surgery. Okay, and for postmortem dissection, we're getting the tissue block from a dead human. Okay, from probably the cadaver, as the case may be. This is tissue sampling, and it's the first step in the paraffin technique. The next step would be considering is fixing. After this tissue block has been gotten, it's good for us to take note of the fact that this tissue block is no longer in a living thing. Okay, so it's been deprived of oxygen. By virtue of oxygen deprivation, this tissue that is made up of cells would gradually begin to undergo autolysis. Now, autolysis is simply a fancy word for self-destruction. Okay, so this tissue would begin to undergo self-destruction, and this self-destruction is due to the action of lysosomes. Okay, so we have lysosomes in the cell. These lysosomes are organelles in the cell, and they are generally enclosed by a membrane that hold all their destructive enzymes in place. So we have cells, we have lysosomes. These lysosomes have these destructive enzymes, but when the cell is leaving, these destructive enzymes are kept enclosed away from the cell itself. However, in the face of oxygen deprivation, when this tissue block has been gotten, these enzymes are released, and it could lead to self destruction of this tissue. Okay, apart from this autolysis, other forms of postmortem degradation could take place. Okay, so this tissue block could start to decay, it could start to undergo putrefaction, which of course is another fancy word for decay or autolysis. Now, what fixing does is that fixing helps to retard these processes of autolysis. It helps to kill the bacteria, and it helps to prevent postmortem degradation. And it's important because when we are examining the tissue, or we are examining the cells and the tissues, we want to know how exactly the cells are. We do not want to examine a cell that has been destroyed by putrefaction or decay or any form of degradation. Okay, so we want to examine the cells as they are. So fixing helps to retard all of this decay and autolysis that could take place in the cell, and it also helps to enhance tissue staining. Now, the liquid or the substance, the solvent made use of in fixing is formalin, and as we'd expect, formalin is used for a lot of preservations. When in gross anatomy or macro anatomy, as we have it, formalin is used for the preservation of cadavers. So it's almost expected that it would also be used in fixing for microanatomy or histology. It's good for us to also take note of the fact that histology is sometimes referred to as microanatomy, and we also have gross or macroanatomy. So formalin is used in this fixing process. Formalin is made of about 40% of formaldehyde. Okay, and as we would see, formaldehyde is an aldehyde. Okay, it's an aldehyde, and it's the very first aldehyde with carbon number equals 1. It has the formula CH2O. Okay, now this helps to retard all of those putrefaction. The next step would be considering is dehydration. Right. Now in dehydration, we have our tissue block. Okay, the tissue block has been fixed. In other words, it has been, so to say, processed to retard decay and postmortem degradation, to kill off bacteria and all of that in fixing. The next step is dehydration, and as the name implies, it's simply removal of water. Okay, so we have this tissue block, and we want to remove the water from this tissue block. So it undergoes the process of dehydration. Now, dehydration is a step in the general step of paraffin embedding. So we have a step, paraffin embedding. Now remember that all of these steps we are considering are steps in the paraffin technique. Okay, now the paraffin embedding is simply the process of removing all of the water that we have in this cell and replacing all of this water with paraffin. However, this process cannot take place at once. Okay, it cannot take place directly. And why is this so? This is so because paraffin is insoluble in water, so there is no way in which we can directly extract all of the water and replace all of the water with paraffin. We need a substance that is soluble in another so that we can have one coming out and another replacing. Okay, this is like having a cloth, and you can say that this cloth is filled with soap foam or soapy foam, and you continue rinsing and rinsing with water, such that what we now have in the cloth, instead of soapy foam, is now water. And this is only possible because the soapy foam is soluble in water, and so over time it can be rinsed off and we now have water. This idea also applies for the paraffin embedding. So we need a substance that is soluble in water in order for us to replace water with that substance. Now, what happens in dehydration is that the tissue block is drenched, so to say. The tissue block is, so to say, drenched in alcohol. Okay, now because alcohol is soluble in water, it's able to replace all of the water in the tissue block with itself. Okay, so all of the water in the tissue block is replaced with alcohol in the dehydration technique. The next step under the paraffin embedding is clearing. Okay, from dehydration we have our tissue block, and instead of water in our tissue block, what we have is alcohol, ethyl alcohol. Now, why exactly do we have to remove all of this water, and why is it so important? Now, it's good for us to take note of the fact that the tissue as it is containing water is soft. Okay, it's soft. Now, what we are essentially doing in tissue preparation is, the end result is to get very thin slices of this tissue, very thin slices of this tissue, so that they can be viewed under the microscope. Now, in order to slice this tissue block, we don't want it to be soft. Okay, we don't want the tissue block to be soft, because if it's too soft and then we are trying to slice it into very thin slices, we have chances of the tissue just cutting or whatsoever the case may be. So what the paraffin embedding does is that it, as an end result, helps to make the sectioning easier. Okay, it helps to make the cutting of the tissue easier. Now, in clearing, we now have our tissue block full of alcohol. What happens in clearing now is that this alcohol is replaced by xylene. Okay, so clearing replaces all of the alcohol in this tissue block with xylene. Now, xylene is dimethylbenzene. Okay, xylene is dimethylbenzene, but dimethylbenzene has 3 isomers. Okay, so we have paraxylene, we have metaxylene, and we also have orthoxylene. Okay, so we have our benzene ring that looks something like this from organic chemistry, and we have this methyl group CH3 taking its position at different locations. Okay, so we could have our methyl group here, we could have it dimethyl group here, and then it could take the position of carbon 1 and carbon 3, it could take the position of carbon 1 and carbon 2, or take the position of carbon 1 and carbon 4. Carbon 1 and 3 is metaxylene, carbon 1 and 2 is orthoxylene, and carbon 1 and 4 is paraxylene. But we are not focusing on all of this. The particular xylene structure drawn here is actually orthoxylene. So we can see that the methyl group takes the position of carbon 1 and carbon 2. We are not focusing on all of this. However, it's good for us to take note of the fact that xylene is simply methylbenzene, and because alcohol is soluble in xylene, we're able to replace all of the alcohol in the tissue block rather, with xylene. The next step is the embedding. Okay, it's the last step of the paraffin embedding. So the goal of the paraffin embedding is to replace all the water in this tissue block with paraffin. Okay, however, as I said earlier, this process cannot take place directly. It needs to take place in steps because paraffin is insoluble in water. Now, the last step of paraffin embedding is the actual embedding. Okay, now what happens in this embedding process is that now we have our tissue block full of xylene. What happens in this process is that all of this xylene is replaced with paraffin. Okay, because of course paraffin is soluble in xylene, and so it's able to take its place in the tissue block. So now we have a tissue block embedded in paraffin. What next? Next step in the paraffin technique is sectioning or microsecting. Now, sectioning or microsecting is simply cutting. Okay, what happens in the sectioning is that now we have our tissue block. Okay, we have our tissue block, and it's not too soft, and it's neither is it too hard, but the paraffin embedding makes it so that we can cut this tissue block easily. So what happens in the microsecting or the sectioning is that we now first of all scrape off or trim away the surplus wax. Okay, paraffin is wax as it is, and in sectioning, first of all, the excess wax on the surface of our tissue block is first of all trimmed away. Then we use a special device called a microtome. A microtome is used in slicing. Okay, it's used in slicing this tissue block into very tiny slices, and it's just about 5 to 8 micrometer of thickness. Very slim and very thin slices of this tissue is gotten, and the next step in the paraffin technique is staining. But before we consider staining, let's take a look at how the microtome looks. Here we have a picture of the microtome, and we can see that we have here the block holder, the tissue block holder, and we have the knife holder and the base with the knife. So this is just a diagram, a picture of how the microtome looks, and this is the machine used in cutting the tissue block into very slim slices. Okay, so it can be viewed under the microscope. The next step in the paraffin technique, which is step 4, first we consider tissue sampling, then fixing, then paraffin embedding, which had about 4 steps, and then sectioning, and now we are at step 5, which is staining and mounting. Now in the staining step, we no longer have a tissue block. What we now have is this very slim sheet of tissue slice. Okay, we have this slice of tissue. Now in staining and mounting, we now want to place this slice of tissue, very slim slice of tissue, on a slide. We want to place it on a slide, and we also want to cover this tissue slice with a cover slip. Alright, so now we have a tissue slice, and this tissue slice has been cut such that if it were to be studied under a light microscope, light can easily pass through it and there would be no overlapping structures, so we can clearly examine the tissue. Now, however, the tissue components and the cell components have relatively close optical density. Okay, now we are studying this under light microscopy. So these tissue components have relatively close optical density, and by virtue of having this close optical density, it means that the degree to which they refract light is very similar. Okay, so without staining, this tissue slice would look like a tissue slice. It would not display the components as they are to be viewed. Okay, so we won't be able to examine the tissue components clearly, Now this is where staining comes to play. Now in staining, we make use of certain dyes and certain stains that help to increase the optical density of these tissue components, making them to be more visible under the microscope. Okay, so in staining, of course, we have different stains. Now after this tissue slice has been stained, it is stained very interestingly. Okay, so now remember that we have this tissue slice, and this tissue slice is, of course, paraffin embedded. It has been paraffin embedded from the previous steps that we had in the paraffin technique. However, these stains and these dyes that we have are not soluble, so to say, in the stains, or the stains are not soluble in the paraffin. Okay, the stains are not soluble in the paraffin. So how exactly do we stain this tissue slice? Now this is very interesting because we would have to reverse the entire process of paraffin embedding. So in paraffin embedding, we had a tissue block full of water, or with water, and then we removed all of the water and then we embedded it in paraffin. Now what happens in the staining process is that this step is reversed. So we remove the paraffin now and basically replace it with water in order to stain this tissue slice. Now after it's replaced with water, the tissue is appropriately stained with the right dyes and stains, and then after the staining is done, the tissue slice is placed on the slide and a mounting medium is applied. Okay, so we have a mounting medium. Now what is the function of this mounting medium? Of course, we want the stain to help increase the optical density. However, excessive refraction of light could also make the slide kind of blur. So what the mounting medium does is that there's a lot of refraction taking place now. We have refraction from the cover slip, from the slide itself, the glass slide. We also have refraction from the stain, okay, that has increased the optical density of this tissue component. What the mounting medium does is that it helps to reduce the refraction of light. It helps to minimize the refraction of light. And in addition to that, when the mounting medium is applied, when the mounting medium is applied to this tissue slice, and then the tissue slice, of course, is covered with the cover slip, this mounting medium, after evaporating, helps to stick the cover slip onto the slide. And now we have our slide prepared, our tissue has been prepared, and it can now be viewed under the microscope. These are the steps in paraffin technique. Now let's take a look at another sectioning technique. Another sectioning technique would be considering is the frozen section. Now, unlike the paraffin technique where we go through a lot of steps and we go through a lot of processes, the frozen section is used when we want to study the tissue immediately. Okay, so it's used for an immediate study of the tissue or the tissue block. So what happens in the frozen technique is that it doesn't undergo all of the whole fixing procedure and all of those things. The tissue block is simply frozen. Okay, so we have our tissue block, and this tissue block is frozen for two reasons. Frozen to retard the postmortem degradation. So instead of fixing, what we have is that the tissue is frozen, and apart from, of course, reducing the postmortem degradation by freezing, it also helps to make the tissue block to be cut easily. Now you say, well, how is this tissue block frozen? This tissue block is not frozen in a refrigerator. It's actually frozen with liquid nitrogen. Okay, now from general chemistry, we know that liquid nitrogen is a good refrigerant. Okay, now apart from being used in a refrigerator as a refrigerant, the liquid nitrogen is also used in histology for frozen sections, and it freezes the tissue block almost instantly. Okay, now instead of using a microtome as in the paraffin technique, what we use in the frozen section is a cryostat. Okay, now the cryostat helps to keep the cutting knife at a low temperature, so it helps to maintain the low temperature of the tissue block, and it slices right away. Now the tissue blocks or the tissue slices have to be cut slightly thicker. Okay, so the tissue slices in the frozen sections are cut slightly thicker than in the paraffin technique. Another type of sectioning is the semi-thin sectioning. The semi-thin sectioning, as the name implies, is a semi-thin section. Embedding medium used is not the paraffin, it's either epoxy or acrylic resin. Okay, and for staining in the semi-thin technique, we make use of toluidine blue. Okay, so toluidine blue. And remember, in the paraffin technique, the thickness was about 5 to 8 micrometer. However, here what we have is about 0.5 to 2 micrometer. Okay, and from our outline, the next subtopic is light microscopy. So light microscopy simply involves the use of the light microscope to study tissues. Okay, there are two terms we should be familiar with in light microscopy, and these terms are magnification and resolution. Now, put very simply, magnification simply has to do with enlarging. Okay, so in magnification, we simply enlarge the image. Okay, so the image is enlarged, is made bigger so we can see it clearly. However, resolution does a different thing. So while magnification simply produces an enlarged image of a small object, so we have a small object and it enlarges it and we now have a big object. Resolution helps to reveal and disclose very fine details. As we would go on to see, in a certain structure you could have two details, so you have detail 1 and detail 2. Now, these details have happen to be very close together. Okay, so we have this detail 1 and detail 2, very close together. Now, in resolution has to do with the ability for us to be able to distinctively identify detail 1 from detail 2. Okay, so if we have this, maybe in a structure we have box 1 and box 2. Okay, resolution is the ability for us to be able to identify box 1 separately from box 2 in spite of their close proximity. So magnification would simply enlarge the image, making it bigger, but resolution will help us to be able to see distinctive details, distinctive features of the structure. Light microscopy has a limit of resolution, and the limit of resolution for light microscopy is 0.2 micrometer. So if we have two details or two distinctive entities, detail 1 and detail 2, if the distance between these two details happen to be less than 0.2 micrometer, okay, the light microscope would be unable to represent these entities as separate. Okay, so they would just simply appear as though they were one entity, it won't be able to distinct this image. Of course, the microscope would magnify the image and make it bigger, however, won't be able to tell whether the box is a single box or it's actually made of two compartments, as the case may be. Okay, now let's take a look at some parts of the light microscope. Here in the picture of the light microscope, we can see that we have different parts of the microscope. We have the ocular lens, which is where we look into. Okay, so we have an observer here looking into the optical lens to see the magnified image. Then we have here the revolving nose piece. The revolving nose piece helps us to select our desired objective lens. Okay, so we have different objective lens of times 10, times 40, as well as times 100. Now the times 100 objective lens is also sometimes referred to as the oil immersion objective or objective lens, and would see the reason why this is so in a bit. However, the times 10 objective lens is referred to as low power objective, times 40 is referred to as medium power objective, and or some, as the case may be, high, and times 100 is a high power objective. Okay, now the nose piece helps us to select either one of these objective lenses. We also have the stage, which has stage clips that help to hold the slide. For this particular microscope, it has a mechanical stage. By being a mechanical stage, it means that it has a stage controller or the stage adjustment. Now this stage controller helps us to move the stage or to move the slide backward or forward, left or right, okay, in order to, so if we have we have the slide this way, and we have the cover slip. Okay, the cover slip basically covers the actual section of tissue to be viewed. Now it's possible such that the slide is placed and the tissue to be viewed is not directly on the aperture. The light is not exactly getting into the tissue, or the tissue is not being able to be viewed from the optical lens. So we could use the stage controller to adjust the position of the slide from left to right or top to bottom, as the case may be. We also have the coarse adjustment knob. So when we have the objective lens on the low power of times 10, the coarse adjustment knob is used to move the stage up or down in order to focus at low power magnification. However, at high power magnification of times 40 or times 100, we usually have the stage very close to the objective lens, so we won't be able to use the coarse adjustment knob, because the coarse adjustment knob actually moves the stage, and further movement of the stage may actually lead to the objective lens cracking the slide. Okay, so when the objective lens comes in contact with the slide, it's likely to crack it. So what the fine adjustment knob is used for focusing the object at high magnification. So at magnification of times 40 and times 100, it helps to focus the slide. Okay, we also have here the base, which supports the arm and the head of the microscope. We have the diaphragm and the iris, as well as the condenser here. These help to direct the light, control the amount of light, and also allow for the entrance of light into the aperture of the stage. Then we have the light source, also known as the illuminator here, and the arm of the microscope. So these are the basic parts of a light microscope, as we would see. When considering the times 100 magnification, we mentioned that it's sometimes referred to as the oil immersion magnification. Now, why is this so, and why exactly do we have to make use of oil for the times 100 magnification? Now, as I stated earlier, usually at times 100 and times 40, the objective lens is always very close to the slide, and I stated that further movement of the stage might lead to the cracking of the slide because the objective lens would meet with the slide and crack it. Okay, now because of the close proximity of the objective lens with the slide at times 100 magnification, the accommodation for refraction is somewhat reduced. Okay, so from physics we know that a certain object distance is needed in order to create a certain magnification. So object distance affects image distance and in turn affects magnification. Okay, however, all of this is made possible by refraction. Okay, so we have a lens and we have the object distance u. It produces an image and the image is magnified. Okay, however, this refraction is because the light is going from air, which is of a lower optical density, and it's going from air into this glass. Now, because the objective lens is very close to the slide, the oil immersion or the immersion oil is applied here. Okay, so it's just applied on the slide where the cover slip is, usually where the cover slip is covering the tissue slice. It's applied there in order to reduce refraction. Okay, so as we can see in this image, it helps to get rid of this air gap. Okay, so it helps to get rid of this air gap. So now occupying this air gap would be the immersion oil. Okay, reducing the refraction. So instead of the light going this way and this way before getting into the objective lens, because the oil has a similar refractive index as the glass, the light travels through the objective lens and it helps to produce the required magnification. That is why times 100 is referred to as the oil immersion magnification or objective. Another thing for us to take note of is what we call the field of view. And it's good for us to take note of the fact that the field of view decreases as the magnification increases. Okay, so at high magnification, we have a smaller field of view, and at low magnification, we have a higher field of view. What does this mean? Okay, so given a particular tissue slide, at low magnification, we are able to view, take for instance, the entire of the tissue slide. So we are able to see this entire structure. This is the field of view. Okay, the field of view. And at low magnification, depending on the size of the tissue slice, we might be able to see the tissue slice in full view. Okay, in full view. However, as the magnification increases, say at times 40, the field of view is reduced such that now we might only be able to see this section. Okay, the field of view is decreased. However, the resolution and magnification is increased, so we are able to see more details. However, the field of view is reduced. Okay, it's like, take for instance, you have a picture. Okay, so you have a picture of any picture in general. You have a picture of a human here. Now this picture is enlarged, however we are still able to see the entire portrait of this human. Now at higher magnification, take for instance, we zoom into this picture, and so we zoom to take a look at how the person's eye looks like. Now we are able to produce a magnified view of the eye. However, we are no longer able to see the entire portrait. Okay, this is what field of view entails. So as magnification increases, the field of view decreases. Now let's take a look at some of the histological stains. Now, as I said earlier, given the tissue slice, so we have the tissue slice, most of the tissue components have very similar optical density. So without staining, they would all appear the same, and we won't exactly be able to differentiate one tissue component from another. And this is why tissue staining is important. Okay, so there are various dyes and various stains used in tissue staining, and it's essentially to increase the optical density. However, this applies to light microscopy. Okay, so we refer to optical density when using light microscopy. However, for electron microscopy, we make use of electron microscopic stains. Okay, so electron microscopic stains. The purpose of these electron microscopic stains is to increase the electron density. Okay, so for electron microscopy, rather than increasing optical density, what we have is an increase in electron density that is achieved by electron microscopic stains. Okay, now let's take a look at some of the dyes made use of for histological stains. In a given dye or histological stain, we have the color impacting component. What exactly is the color impacting component? Now, the color impacting component is that component of the dye that gives the color. Okay, now in the dye we have different substances, a mixture of different substances. However, a particular component of the dye is actually what impacts the color and not the entire of the dye. Now in a given dye we could have an acidic color impacting component, or we could have a basic color impacting component. Now as the case may be, this dye, or generally the dye, what the dye provides is a bright color stain. Okay, and we also have what we call a counter stain. Now what this counter stain does is, not all the components are colored by the dye. Okay, so not all the components receive their color from the dye. So the counter stain helps to give color to those other components that have not received the color stain. Okay, and it usually gives a contrasting color. While the dye generally gives a bright color, the counter stain gives a contrasting color to the remainder of the tissue components that have not been stained. For this video, the particular stain or dye would be considering is hematoxylin and eosin. Taking a look at the H&E stain, or the hematoxylin and eosin stain, the hematoxylin part of this stain gives a bluish purple color. It gives a bluish purple color to the tissue component, while the eosin gives a pink to red color to the tissue component. It's also important for us to take note of two terms. These two terms are acidophilic and basophilic. Now, philic simply means loving. So acidophilic would be acid loving, and basophilic would be base or alkaline loving. Now, in a given cell or in a given tissue, okay, we have certain components of this tissue that are acid loving. Okay, so these tissue components love acid, they are attracted to acid and attracted by acid. We also have other components of the tissues that are base loving. So these tissue components are attracted to the base and the base is attracted to them. Hematoxylin is a basic stain. What does this mean? Being a basic stain means that the color impacting component of hematoxylin is a base or a basic radical. Okay, this simply means that the basic radical reacts with OH and then impacts a stain onto the tissue component. So hematoxylin is a basic stain and it colors basophilic tissue component. Okay, so a basophilic tissue component is base loving and it's attracted to this basic stain. The basic stain has the color impacting component as a basic radical. Now, of course, we know that if we have a typical base, take for instance NaOH, the basic radical is Na, which is sodium ion, and it's a positively charged ion. Okay, now this tells us a bit of an information about the tissue component. Okay, so given a tissue component such as this that is basophilic, okay, and it's basophilic because it is colored by a basic stain. However, the color impacting component of the basic stain is a basic radical, such as, take for instance, sodium ion. Now, sodium ion is positively charged. It means that for sodium ion to be attracted to another substance or to another component, that component would definitely be negatively charged. Taking a look at eosin, eosin has its color impacting component as an acid radical. Now, taking a typical acid, for example HCl, the acid radical for this acid would be chlorine ion, which is negatively charged. Now, eosin stains the acidophilic components of a tissue. Okay, so the acidophilic components of a tissue are stained by the eosin, which is an acid stain. Now, given that the color impacting component is an acid radical which is negatively charged, this tells us a bit of an idea that the acidophilic component that is stained by eosin is somewhat positively charged since it's attracted to the negatively charged acid radical. Okay, all of this staining and all of these colors that we have for hematoxylin, we have a bluish purple stain. Okay, so bluish purple, and for eosin we have a pink to red stain. Now, all of these stains and colors that we have are because of the nature of light. Okay, now from physics we know that white light is made of a spectrum of colors. Now, because light or white light is made of this spectrum of colors, by refraction and diffraction we are able to actually see these colors under the microscope. However, in electron microscopy, in electron microscopy we make use of electron beams, and these electron beams, unlike light, do not have a color spectrum. So what do we expect? This simply means that the photomicrographs of a light microscope give us colored images. So we have colored images from a light microscope, and photomicrograph is just a picture of a small image, a picture of essentially what is viewed under the slide. And photo because it's from a light microscope. Micrograph, the picture of a very small image. Now the photomicrograph is a colored picture. Take it as a colored picture. However, as I was explaining for the electron microscope, what is used is electron beams, and these electron beams do not have a color spectrum. So as expected, the micrographs from an electron microscope would be black and white. So we have them in black and white, and this is because the electron beam doesn't have a color spectrum. Picture of a tissue slide that is stained in hematoxylin and eosin. So we can see some purple and a bit of, I can't really see pink and red, but I can see the purple appearance from the hematoxylin. Now from our outline, the next subtopic is histological sections. So let's take a look at some of the histological sections. When we have a tissue sample that is of greater length, as we have here, this tissue sample, the structure representing the tissue sample here has a greater length than width. So when a tissue sample such as this one is cut along its longer axis, such a section is a longitudinal section. Longitudinal section. When it's cut at an angle such as this, it's an oblique section. When it's cut perpendicular, horizontally at right angle, it is a transverse section. Round tissue samples or round shapes, we could also have a transverse section horizontally at 90 degrees, such as this one, and we could also have a tangential section. A tangential section simply grazes over the surface. It just acts as a tangent would touch a circle just at one point. This tangential section also simply grazes over the surface of the tissue sample. Next subtopic is artifacts. Now, artifacts are simply flaws. They are flaws that are caused by faulty techniques. Now, in this video so far, we have considered paraffin technique as a technique of tissue preparation. However, certain flaws are likely to occur either by faulty handling or faulty technique, and some flaws are just simply unavoidable. Now, when viewing a particular slide, certain artifacts can be seen. Now, these artifacts are not part of the tissue, so to say, simply artificial structures that form as a result of faulty techniques of tissue preparation. On our list is postmortem degeneration. Now, postmortem degeneration results from unavoidable delay in tissue preparation. So, as I stated earlier, the tissue is now deprived of oxygen. It's deprived of oxygen, and due to these lysosomal actions, it undergoes putrefaction, bacteria works on it, and a lot of breaking down occurs in the tissue because it's no longer living. Now, postmortem degradation could lead to some of these artifacts because now the tissue is undergoing autolysis and the structure is, in other words, distorted. Okay, this is one of the artifacts we'd be considering. Another artifact we'd be considering is shrinkage. Now, because of the reagents used for the preparation of a tissue or tissue preparation, some of these reagents lead to shrinkage. Okay, and an uneven shrinkage. An uneven shrinkage generally leads to cracking. Okay, so we have cracking in this tissue components due to the reagent. Now here we can see in this tissue slide, this tissue slide is the epithelium of the jejunum. Okay, the epithelium of the jejunum, and it is a simple columnar epithelium. However, it appears as though it's a stratified epithelium. It's important for us to take note of the fact, however, that these empty spaces, the illustration of the empty spaces that we can see in this slide, are actually artifacts. Okay, if we take a look at this part of the epithelium, for example, we can see that it actually forms a column and it's directly attached to the basement membrane as a simple columnar epithelium. However, the cracks and empty spaces we can see here is an example of an artifact, a shrinkage artifact that is caused as a result of cracking in faulty techniques of tissue preparation. Next type of artifact we'd be considering is precipitation. Now, when we were discussing the paraffin technique, we saw as one of the steps in the paraffin technique fixing. Now, in fixing, we saw that the substance made use of is formalin. Now, formalin has the ability to precipitate in these tissue sections, and an artifact is formed when formalin forms precipitates in tissue sections. Now, particularly in tissue sections that contain hemoglobin or red blood cells, acidic formalin has the ability to react with hemoglobin to give this tissue section if a sort of granular appearance. So we would see, like in the tissue section that we have here, we can see this granular brown structures. This is as a result of formalin precipitation. Another artifact is wrinkle or folding. Now, this occurs when the tissue section is being placed on the slide. Okay, so we have a tissue section, which is basically a slice of tissue, and this tissue section is to be placed on the glass slide. However, in trying to place this tissue section onto the glass slide, the tissue section may wrinkle or fold, and this wrinkle or fold would appear as an artifact under the microscope. This tissue section exhibits another type of artifact, which is formed or caused by the microtome knife, and it's also called the knife nick artifact. Now, a nick is simply a cut or a notch, and we can see here in this tissue sample that in the process of sectioning using the microtome, we can see here that the knife of the microtome has sort of cut into the tissue section such that we have these cuts in the tissue. Okay, now these cuts are not part of the actual tissues, but they are artifacts that have been formed as a result of the microtome knife nick. Okay, certain part of the tissue is held by forceps, and certain parts are cut by dull scissors. Now, this two instruments could cause some artifacts in the eventual tissue section that is prepared. Okay, so these are some of the unavoidable artifacts that can be created during tissue preparation and tissue sampling. Next, let's briefly take a look at electron microscopy. This video would consider electron microscopy very briefly. Now, as earlier stated, in the light microscope, we're making use of light. Okay, so light is directed at the tissue section in order for it to be viewed. However, in an electron microscope, the electron beam is either transmitted through the tissue section or it scans through the tissue section. This gives us a transmission electron microscope, EM, as well as the scanning electron microscope. Now, in the transmission electron microscope, the electron beam is transmitted through the tissue section, so it goes through the tissue section. However, in the scanning electron microscope, the electron beam simply grazes over the surface. It scans over the surface of the tissue section and it's not transmitted through it. And this is because generally, from our knowledge of physics, we have something that looks like this. Now, what we have here is the penetrating power of the three types of radiation. And in an electron microscope, we make use of the electron beam, as stated earlier. Now, this electron beam is very similar to the beta radiation. Okay, and here in this picture, we can see the beta radiation. Okay, now taking a look at the beta radiation right here, we can see its penetrative power. Now, the beta radiation is able to pass through a sheet of paper, however, is unable to penetrate through a sheet of aluminium. Now, as I stated earlier, the electron beam is similar to the beta radiation. And because the electron beam has a relatively low penetrating power, it means that the tissue slide or the tissue section must be extremely thin. Okay, it must be extremely thin in order for the electron beam to be transmitted through it. However, for the scanning electron microscope, the beam is not transmitted through the section. Instead, it just scans the surface and gives an image of the components of the tissue components on the surface. Now, for an electron microscope, an ultra microtome is used for its sectioning. Okay, an ultra microtome is used for its sectioning, and unlike the light microscope, where in the paraffin technique we made use of about 5 to 8 micrometer of thickness, in the electron microscope about 60 to 80 nanometer of thickness is required. So the sections are indeed extremely thin as compared to the light microscope. Also, it's important for us to take note of the fact that because the electron beam doesn't have a color spectrum, the micrographs produced by an electron microscope are in black and white. Okay, they are in black and white. Now, why exactly is the electron microscope so important? When we consider the light microscope, we saw that we have the objective lens of times 10 magnification, times 40, and times 100. Something I forgot to mention is that the eyepiece has its magnification. The eyepiece lens has a magnification of times 10. Okay, now in order to get the total magnification for a tissue section or for a tissue sample, it would be the objective lens magnification multiplied by the eyepiece lens magnification. So if we are making use of the objective lens of times 40, it would be 40 times. The objective lens always has a magnification of times 10, or usually has a magnification of times 10, so it would be 40 times 10, and the total magnification would be times 400 for the tissue section. Okay, now the highest magnification that can be produced by an electron, the highest magnification that can be produced by a light microscope is times 1400. However, an electron microscope is able to produce a magnification of up to times 50,000. Okay, now this simply means that we are able to see finer details of a given tissue sample, and it also means that since more details can be seen, means that it offers a higher resolution. Okay, now in speaking about resolution, stated earlier that the light microscope has a limit of resolution of 0.2 micrometers. 0.2 micrometers. In other words, if we have two separate entities such that the distance between these two separate entities is less than 0.2 micrometers, it means that the resolution of the light microscope would be unable to distinct these two images. So these two entities or these two details will appear as though they were one in a light microscope. However, the resolution offered by an electron microscope is much higher. So even if the two entities had a distance far less than 0.2 micrometers, they can still be identified distinctively. Okay, so these are some of the importance of an electron microscope. Also, unlike in the light microscopy where we make use of stains and dyes, in the electron microscopy we make use of heavy metals. Heavy metals are used as the stain. Okay, heavy metals are used for electron microscopic stains, and this is because they help to scatter the electron beam and so can produce an illustration of electron beam of electron dense areas and areas that are less electron dense. So in summary, the electron microscope is necessary for the observation of very tiny structural details. We have come to the end of this video. If you liked this video, please give it a thumbs up and don't forget to subscribe. Thank you for watching till the end. Bye.